In the last lesson on the concept cells, we asked the question, what happens to the response properties as you move down the visual pathway into the temporal lobe, but then enter the hippocampus? We saw that as you go from primary cortex down through the temporal lobe, the cells start to have a preferred stimulus that's more and more complex, but they no longer care about the color or the size or the motion or the texture of the object, really just sort of the shape, the identifying shape of the object. So the question then was, what happens if you continue on this kind of visual hierarchy down into the hippocampus, remembering that the hippocampus integrates perceptual information from the different regions of the brain? Well, the idea then would be to record from hippocampal cells while showing patients different kinds of objects and try to find the preferred stimulus. And we saw that was the strategy in animals in recording from fusiform cells. Here we're going to try to record from hippocampal cells. Now, how do you do that in humans? Well, again, it's epilepsy patients who are sort of volunteering themselves for science. So in order to treat severe epilepsy, scientists first try to record electrical activity in the brain to find out the locus of the seizures. And sometimes the electrodes are inserted into interesting areas like the hippocampus. And in one of these cases, when a patient had an electrode inserted there, scientists took advantage and began to try to test the neuron's preferred stimulus. And here's one of those famous neurons discovered a few years ago. It's the Jennifer Aniston neuron. Now, the little bars here indicate the responses of the hippocampal neuron to various visual stimuli. For all of these pictures, which are all Jennifer Aniston, the neuron shows high activity. And for all kinds of other people and places, generally speaking, the neuron shows very little activity. In other words, this cell was selective for Jennifer Aniston. What's interesting is that these pictures show Jennifer Aniston in different positions, different angles, different hairstyles, different clothing, different background, but the cell didn't care. It was firing to the Jennifer Aniston picture. So there's sort of an invariance there. And it's kind of reminiscent of some of those inferior temporal cortex neurons that don't care about color or size or whatnot, just the shape. And this cell seems to care only that Jennifer Aniston was in the picture. Now here are some cells that are responding to Luke Skywalker, to Mark Hamill. So we have activity here for all these conditions. Oh, but now take a look at this. The cell not only responds to the face of the person, but also the written name of the person, Luke Skywalker, and beyond that, the auditory experience of hearing somebody say the name Luke Skywalker. So here we have a multimodal response that this hippocampal neuron will respond to the visual stimulus of Luke Skywalker, the written word Luke Skywalker, and somebody saying Luke Skywalker. So now we're starting to see, unlike the inferior temporal neurons that are sort of in the visual pathway, this hippocampal neuron can be activated by auditory stimuli and written, that's visual also, but of a different kind than a picture. So these researchers began to characterize these hippocampal cells as concept cells. So selective responses. So they only responded to a fraction of the presented pictures. There might be 100 pictures presented, and they just respond to a fraction, like the Jennifer Aniston or the Luke Skywalker. The responses show invariance to specific visual features. Didn't matter the visual angle of Jennifer Aniston or Luke Skywalker. Didn't matter the size, the clothing, hairstyle, the background, and whatnot. They were multimodal responses. So not only to pictures of the people, but their written name and their spoken name. And sometimes, and actually often, these concept cells did show some activity to other stimuli, but those stimuli were usually related concepts. So for example, take the Luke Skywalker neuron here, it did respond to Yoda. And in fact, you don't see it here, but in one of the Jennifer Aniston neurons they found it also responded to pictures of Lisa Kudrow. Now, these concept cells what was interesting also is that neighboring cells respond to different concepts. So there's not a topographically organized system in the hippocampus. It's not like two neighboring hippocampal cells were Jennifer Aniston cells. You might find a Jennifer Aniston cell next to a Halle Berry cell, next to an Oprah Winfrey cell, for example. These researchers also suspect that we do here have a case of sparse coding, that relatively few neurons are active for a given concept. And I think this is possible because typically humans don't remember many more than around 10,000 concepts. So that puts an upper limit on the number of cells you would need to code for distinct concepts. But more than that, some of the theoretical modeling suggests that sparse coding has some advantages for making fast associations between newly learned stimuli. And so what they think is going on is that these concept cells are encoding the meaning of a stimulus for memory functions, like making associations with other concepts and contexts. And in their view, sparse coding is a more efficient system for a rapid association learning system. Now that you'll recall the sparse coding versus distributed coding. In distributed, you have relatively more cells, and what differentiates concepts would be the relative activity of those cells in the network. In sparse coding, it's fewer cells, and they're very selective for different concepts. So while it may be that we have evidence that the fusiform face area is using distributed coding, the population coding, that doesn't mean that every brain system uses that type of coding. And it could be that in the hippocampus and other related structures, it's sparse coding. In an earlier lesson, we learned about some animal research that was asking the question, do the same cells that are processing a cue, do they get reactivated when that stimulus is retrieved during memory retrieval? And recall that we called this the reactivation hypothesis, that maybe the same areas of the brain that are used for perception might get reactivated when we call up a memory. Well, the team here that was recording from hippocampal cells used this opportunity to sort of test this idea, the reactivation hypothesis. And here's how the experiment went. They would have the subject looking at a screen on which they played lots of little video clips, and then they were simply sort of recording from cells in the hippocampus and other medial temporal lobe areas, but hippocampal cells, while they were watching, they wanted to find hippocampal cells that responded to certain segments of a video clip. And then they would have some kind of intervening task, so they want to create a delay here, and then at a later point they simply ask the subject, what do you remember seeing? Now notice, that's a very sort of just general cue to reactivate an earlier memory, but there is no external cue itself. It's just, what do you remember seeing? And what was of interest to the researchers was, if a hippocampal cell responded to a certain video clip, would that cell reactivate when the person was retrieving the earlier memory of that video clip? The best way to sort of see this is to see the video of it. So I'm going to grab this off the internet. This was by Itzhak Fried and his colleagues. All right, as it turns out, in this particular case, they're recording from an entorhinal cortex neuron, and the entorhinal cortex sends its axons right into the hippocampus. So here we go. The record of the entorhinal cortex neuron is down here. So they're recording from this neuron, and they want to find a video clip that will show increased activity in that neuron. Notice they're showing different kinds of video clips. Here's a Titanic clip, and we already see that the entorhinal cortex neuron is going to spike here. What video is it? The Simpsons. So they found that the Simpsons video clip showed a big spike in activity for that entorhinal cortex neuron. Now, they wanted to cycle through the video clips several times to see if this was repeatable. Is it that cell is selectively responding to the Simpsons? So they go through some more video clips. Here comes the Titanic scene again. The King video, and now the Simpsons. So at this stage, they've characterized this entorhinal neuron, and then they have a delay, so they do something else, have a little chat or whatnot, and then they're going to ask the subject to simply remember something from the videos that they saw before. So they're not giving them any specific cue. The question is, will that same entorhinal neuron show reactivation if the subject remembers The Simpsons? So here's what they said. So at this point, they said, oh, they remembered something about New York. But the entorhinal neuron did not show a spike in activity. The subject said the Hollywood sign they remember seeing in one of the videos, but again, no elevated activity. And you guessed it. So when we saw that the spike in that same cell activity, that's when the person then said The Simpsons. It's as if the activity in this same entorhinal cortex neuron was being reactivated during the retrieval process. When the subject was retrieving the memory of The Simpsons, we get reactivation of that same neuron that was active when the subject saw the Simpson video. So this definitely seems to support the reactivation hypothesis. So perception results in stored representations of the experience, and we can think of them as memories. The hippocampus is instrumental in making an original memory trace of the learning episode. The hippocampus and surrounding tissue, like entorhinal cortex. And so retrieving the memory reactivates those hippocampal and other associated cells that process the episode. So this is the idea of the reactivation. The cells were active when the Simpson video clip was happening, and then showed activity when the memory was retrieved.